Hello, and thank you for clicking on the Literacy Volunteers of Harrison County YouTube channel. We're a nonprofit United Way agency, and we bring these videos to you for educational purposes. We would love for you to subscribe to our channel. Just click on the subscribe below. If you hit the bell icon, you'll be notified when we put up new videos. Today, our video is on genetically modified organisms, or GMOs. So the first thing we want to ask are, what are they? What are GMOs? They are living things that have had their genetic code, the actual DNA, changed in some way. So we're going to look at how that occurs. The G stands for genetic, right? That means they're made of DNA. If you look for DNA, it's found in the nucleus of all of your cells, of each of your cells, of every living organism. It carries the instructions that control how the cell grows and develops. So it sort of makes you you. You get half from your mom and half from your dad, right? Or any organism does, and then that's your genetics. M stands for modified, and modified just means that we've changed it some way. When we modify something, we change it. And then O stands for organism, and all living things, plants, animals, bacteria, fungi, those are considered organisms, each individual one. So let's look at a little bit of the history to see kind of how GMOs came to be. In 1973, we had these two biochemists. One's name was Herbert Boyer and the other one was Stanley Cohen. And what they did was they developed genetic engineering by inserting DNA from one bacterium into another. And we can do that. I've done that with my biology kids in the lab. You take what's called a plasmid. And a plasmid is just a genetic structure in a cell that can replicate independently of chromosomes, which means it can replicate on its own, right? It's usually small, it's usually circular, like that, and it's a DNA strand found in the cytoplasm of a bacterium. So what we would do with this plasmid was, if we had a gene that we wanted, we could excise it or cut it out. We remove this gene and we can insert that gene into a different bacterium. This is to represent E. coli. It could be any bacterium. Bacteria do not have a well-defined nucleus. We've talked about those. They're prokaryotes. So they have, their DNA is just a circular DNA, and it's usually attached somewhere to the coat. So we can put that into this organism, which is what they did. And they're used in labs, these plasmids, to manipulate genes. Take them out, put them in, move them around to see how we can change them. In 1982, the FDA approves the first consumer GMO product that was developed through that genetic engineering process, and it was human insulin. And we use insulin to help treat diabetics. So they made this human insulin in a lab so that they could treat diabetics. In 1986, the FDA, the Food and Drug Administration, the EPA, the Environmental Protection Agency, and the United States Department of Agriculture worked together to regulate the safety of GMOs. People didn't think that they were safe, so what they have to do is they all have to come together to try to figure out how to make them safe for the consumer. So by 1994, the first GMO produce was created through genetic engineering, and it was a GMO tomato, and they put that up for sale. So it took all of those years, 21 years, to come from when we started moving genes around in a plasmid to when we create this genetic GMO tomato. And in 2003, the WHO and the FAO plus the United Nations, they come together to develop guidelines. They don't want countries just making things however they feel, so they have to follow these safety guidelines and standards to make genetically modified organisms safe. In 2015, the FDA approves an application for the first genetic modification of an animal. So we've moved here from plants to animals, and this was a salmon. It was a genetically engineered salmon. And so now we have the first animal that has been genetically modified. In 2016, Congress passes a law requiring labeling for some foods produced through genetic engineering and uses the term bioengineered. So when you find those foods, it was stamped on there, this food has been bioengineered, right? It's been changed. Bio means life, engineered, we moved it around. And in 2019, just a few years ago, the FDA completes consultation on the first food from a genome-edited plant, and we're going to look at that here in a minute. So let's look at how we do this. We know what GMOs are now. We've looked at the history. How do we genetically modify an organism? First of all, what we have to identify is what trait are we looking for. When we talk about traits, traits are passed on in your genes. Like if we look at humans, hair color, eye color, height, weight, skin color, all of those are traits. They're individual traits that are carried on your DNA. If we look at, in this instance, corn, we might say the color of the corn, how many kernels there are, how tall it grows, the color of the leaf. All of those things are governed by specific genes. So we're going to pick a trait. What trait do we want? Then we have to find an organism that has that trait. Right? So we have to pick one of those. And then for this example, we're going to look at Bt corn, and it's called that because there was a gene in soil bacterium that's called Bacillus thuringiensis, and that's the B and the T, and that produces a natural insecticide, which means it fights off insects. So what we want to do, we're going to take this gene from this bacterium and place it into corn, because we want, when that corn grows, to have a natural insecticide ability. So we've found our trait, we found our organism, so what do? We take it out and we're going to make a copy of that gene, right? We can do this in a lab, we can make a copy. Then we're going to take that copy and we're going to insert it. These scientists put that gene into the DNA of that plant. Now we have insecticide-resistant corn, Bt corn. That new trait does not change the other existing traits. When we put that trait into that organism, we don't change the color, we don't change the height, we don't change any of those things, because we've only changed one gene. We haven't changed all of the genes. And then the scientists will grow this new corn. They want to make sure that it has adapted that trait, because if it didn't, there's no point in mass producing the desired trait that we needed. So the plants are grown, they monitor those in a greenhouse, and then they take them out and field test those. GMO plants have to go through an in-depth review and many tests before they are ready to be sold to farmers. And that process may take several years by the time that we've perfected that. So that would be one of our genetically modified organisms. And so with this genetic modification, people always get a little nervous when we talk about modifying DNA or changing DNA, because they really don't understand the process, or they don't understand what DNA does. So we have some safeties and concerns. One concern would be allergies, right? We might get this to happen because those GMO foods contain foreign genes that may cause an allergic reaction. Say we had a strawberry and we wanted that strawberry to be more fruity, and we found a gene in a peanut that made it more fruity. So we take it out of the peanut and put it into the strawberry. Well, now we've got a gene that belonged to a peanut in a strawberry, and people eat strawberries who don't know that gene's in there may be allergic to peanuts, and that's going to cause an adverse reaction. So we have to be careful of those reactions. According to the FDA, researchers who develop those GMO foods will run many tests on those foods to ensure that those allergens aren't transferred from one food to another. So maybe that peanut allergy, whatever gene that contains to make you allergic to that peanut, wouldn't be in the gene that they used to put in the strawberry. That's what they're talking about there. Cancers are another concern. Cancers are caused by what are called DNA mutations. And I gave you a little definition. A mutation is a spontaneous change in a gene. And what we do is our DNA is being replicated, we're making copies of it, and all of a sudden one of those genes changes. We have different types of mutations, and we won't go into those here. It could be a point mutation, one base pair. It could be, a deletion or anything, any different kinds of mutations. And so people fear that eating those foods with added genes may affect their DNA, right? May affect the DNA not only of the organism, but of their DNA. So we have to make sure that none of them do that. How do we identify GMO foods? In the United States now, we have corn, soybeans, canola, sugar beets, alfalfa, cotton, potatoes, papaya, summer squash, and apples. All of those have been genetically modified. Right, all of those are sold, that has been perfected. And so beginning in January of 2022, which is just a few months away, the FDA and the USDA will require food manufacturers to label all foods containing GMO ingredients. So you'll be able to see that right on the label. The label won't say GMO, it'll say bioengineered food, so that you